Next up, we have Kylie Bemis from Northeastern University, who's going to tell us about mass spectrometry analysis.
Hello, everyone. And yeah, I think I was set up pretty well by the previous talk here. So I'm also here talking about mass spectrometry imaging. And if you are buying supplements, and I do from time to time, but there are actually some nice third-party testing facilities. So I buy supplements that have NSF Safe for Sports certification. So for anyone who is interested in supplements, there are a few of them that submit for third-party testing, but otherwise it is unregulated, which is why I do that. Otherwise, you don't know what you're getting. Okay, so hello, everyone. So my name is Kylie Bemis. My background is in statistics. I'm coming to you from Northeastern University, Corey College of Computer Sciences. And so I'm going to talk to you a little bit about scaling spatially aware machine learning for mass spec imaging. So this is going to be a little bit more on the computational side of things. So if that's not your cup of tea, I apologize in advance, but I do hope that this will be useful to any of the more computational people out there, and hopefully still kind of illuminating for everyone else as well. So I've included these couple slides in most of my previous talks. I haven't had much of a chance to use them until now because my previous talks were mostly at mass spec imaging conferences. But now, just to get everyone on the same page, I want to briefly introduce or reintroduce mass spectrometry imaging for anyone who might be less familiar with it. So the basic idea with mass spec imaging is we want to essentially have a molecular microscope to be able to look at the distribution of different chemical compounds, different kinds of molecular analytes across the surface of some kind of tissue sample. So we have some kind of tissue sample. So up on the upper right here, we have a section of a pig fetus. And then we collect mass spectra across the surface of that sample. So just below that, you can see a collection of about 20 mass spectra. In reality, we have hundreds, thousands, potentially even millions of mass spectra. In each mass spectrum, we have the x-axis is our mass to charge ratio axis, so that represents different molecules. The y-axis is our intensity, representing the relative abundance of those different molecules. And so with mass spec imaging, what we can do is if we choose a particular mass to charge ratio bin, we can then create ion images, molecular ion images, showing the relative spatial abundance of those molecules in the sample. And so the challenges for mass spectrometry imaging are that we have potentially hundreds of thousands of these different images because we can have potentially hundreds or thousands of molecules in our samples. So here I just have a selection of a subset of some of the ion images from that single pig fetus section, representing many of the different molecules here. So from a machine learning and kind of AI perspective, and my background is as a computational scientist, as a statistician, so that's where I'm coming from. Some of the primary computational challenges are the increasing mass and spatial resolutions of mass spec imaging technology. So we have much better mass resolution than we did when I first started working with this technology about a decade ago. So we can now have highly accurate masses measured by the mass spectrometers now. We also have much better spatial resolutions than we used to. So nowadays, some labs are getting down to the single micron, single cell level. In reality, that's not often used. Usually we're closer to 50, 20 microns, something like that for more practical purposes. But for some of the bigger labs that are pushing the limits, you can get down to single micron, single cellular resolution with this. And all of that means that we are getting much larger data sets than before. So when I first started working on this, a gigabyte was a large data set. Nowadays, that's a pretty small data set, and we kind of start with tens of gigabytes, and you can get up to terabytes in the size of data sets, depending on your mass and spatial resolution. We also have complex experimental designs. You have to think about a lot of things that you don't have to think about when working with traditional proteomics or metabolomics. So the other half of our lab works on traditional proteomics and experimental design and statistical analysis. And so when coming over to the imaging side of things, you now have to start thinking about how you're laying out your tissues on these slides for imaging, which samples you're putting together on the same slide, pesticides, all of those things will affect whether the different sources of variation for your experiment. And of course, some of the traditional kind of statistical issues arise from this, which is you have really large data sets, but you actually have relatively small sample sizes. So from this kind of imaging data, you can end up with hundreds of thousands, millions of pixels. So it looks like you have a lot more data than you really do, but all of that data might be coming from a very few number of samples. So all of these images, for example, these are all coming from just one subject, one single pig fetus section. So these are some of the challenges. What I'll be focusing on today is a specific spatially aware framework for image segmentation we're using. And first, I'm going to briefly talk to you about the open source software that we do have available for this, in which all of this is implemented. So one of the software that we maintain and develop in our lab is Cardinal. This is an R package. It's an open source statistical software for mass spectrometry imaging analysis. And I have to thank also the organizers for inviting me back here to Stanford, because even though I did my PhD at Purdue University and all my publications from that time are from Purdue, Cardinal was actually largely developed here at Stanford. So I spent 2 semesters at the Canary Center here at Stanford working on methods for early cancer detection, and Cardinal was kind of what I was working on at the time. So we were working on using mass spec imaging to identify what are good candidates to then use in MRI for early cancer detection. So figuring out what molecules could be targeted by MRI to detect cancer early on. So that's kind of also a little bit of where the name Cardinal comes from. Cardinal red for Stanford. It also comes from this this painting. So one of the first MS images that we analyzed was this little oil painting of a cardinal from Graham Cook's lab at Purdue. So a little bit of a story behind where all of this comes from. So it's nice to be back here. So this is a fully featured software for the full workflow of analyzing mass spec imaging data. So importing raw data in IMZML format, visualizing it, preparation, pre-processing, including normalization, baseline removal, peak detection, peak alignment, all of that. But our real focus is on statistics, machine learning, and AI. So we have a suite of different tools for analyzing mass spec imaging data using statistics and machine learning. And some of the things that we focus on with this software is reproducibility. So this is all in an R package, so it makes it easy to reproduce your analyses. If you send your code to someone else, they can reproduce it as well. We have releases every spring and fall on Bioconductor. We focus a lot on trying to make sure it performs very well, that it's efficient. One of my goals as coming from a computational lab is that you shouldn't need access to a supercomputer or an HPC cluster to perform analyses. It's great if you do, but if you don't, I still want you to be able to analyze your data. So that's one of the goals of Cardinal. So that's everything about Cardinal. So the rest of this talk is all focusing on a particular spatially aware segmentation method that has been implemented in Cardinal for the last 8 years. So one of the challenges with mass spec imaging, and the specific goal that I'm going to focus on today is unsupervised class discovery. So this is the context where you have a tissue section, you kind of want to explore what are the different morphologies, what are the different regions within this sample, kind of just discover what is going on within this tissue sample when you don't have any previously known class labels like healthy or disease, or maybe you do, but you're not sure what parts of the sample they actually apply to. And so the goal here is unsupervised machine learning to discover regions of relatively homogeneous chemical composition within a heterogeneous tissue sample. And some of the primary computational challenges with this kind of goal is how to choose the number of segments. So how many different regions are we trying to discover within our tissue? And then how do we actually interpret them? So once we see the different regions that come out of the algorithm, what do those actually mean? What are the different molecules that correspond to those regions? So those are two of the kind of challenges that this method tries to make easier and tries to address. So this is a method called spatial shrunken-centroids. This was originally introduced in Cardinal back in 2016. So you might be wondering why I'm talking about it still. I'll mention that in a moment. But the basic idea behind spatial shrunken-centroids is it's a segmentation method that summarizes the morphology of the sample. It includes data-driven feature selection. So it selects the molecules that are most characteristic for each segment, and that is done using statistical regularization. So the output of this method is a nice segmentation map showing the different regions within the sample, and then also feature selection. So it's going to show the different molecules that are actually driving those different segments. So for example, this is a public data set, from PRIDE. And you can see 3 of the main regions in this. I think that it's a mouse bladder. And you can also see the different molecules that are driving those different regions in the upper right corner here. So why am I talking about a method that we introduced in 2016 in 2024? So implementations change over time. We've actually made a lot of changes to how this method is implemented. And some of those improvements have actually impacted how well this method scales and how easily we're able to interpret the results. So a quick background, I'm not going to go into too much detail on this of how spatial shrunken-centroids works, but it's basically a combination of 2 methods that previously existed. So we are using a spatially aware distance from spatially aware clustering by Alexandrov and Kobarg, and then statistical regularization from nearest shrunken-centroids, which is a method from Tibshirani and Hasty. So the basic idea is we're going to compute a dissimilarity between spectra using a spatially aware distance measure. So we're incorporating the spatial component of the data in our dissimilarity between spectra. We assign segment membership based on that spatially aware dissimilarity. We update the segment centroids based on that and update predicted segment memberships. And then we iterate on this until we converge. So we have 2 kinds of weights. One is just a spatially aware Gaussian weight. So these don't depend on the spectra themselves. They're just a weighting based on spatial proximity. And then we also have what we call the spatially aware structurally adaptive weights. So these are borrowed from bilateral filtering in computer vision and image analysis. So these weights do change depending on the actual spectra. The basic idea is that we want to preserve edges between different regions better. So these weights will adjust based on how similar the spectra are in order to better preserve edges between different regions. So the way this method works is we start by computing spatially aware distances. From that, we compute some spatial discriminant scores. We compute the segment membership probabilities. So what is the probability an individual spectrum belongs to a particular segment? And then this is the part where we do the feature selection. We compute the segment centroids. So what is the mean spectrum for each of the different segments? And compute feature-wise t-statistics. So these are essentially the importance of each of the mass features in determining the segments. Here, this is where the regularization comes in. We shrink the t-statistics based on a user-specified sparsity parameter. And then from that, we recompute the shrunken class segment centroids. And then we iterate all through this until the segmentation is relatively stable. And so we showed back in 2016 that this improves segmentation compared to other existing methods, especially if you're not taking into account the spatially aware information. The most important outcomes of this method are that we are able to select the important molecular features. So for example, the brain here, we can see exactly which molecules are important, which ones are more abundant versus less abundant in the brain relative to the rest of the sample. Same thing for the liver there in blue. And the other thing that comes out of this is the sparsity helps us somewhat to select the number of segments. So that's another of the challenges, is how many segments are we actually looking for? And so we allow empty segments to drop out of the segmentation. And as we introduce more and more sparsity, so more sparsity means we're using fewer features. So we end up with more empty segments, and those empty segments drop out of the segmentation. So as we use fewer and fewer mass features to do the segmentation, we end up with fewer segments. And so this can kind of help us in guiding us to select the number of segments based on when this kind of becomes stable. So there were still some practical challenges that we've been working on since then. So specifically, scaling the algorithm to larger than memory data sets is not trivial. That's always a challenge with any algorithm, but it's especially a challenge with spatially aware algorithms because you have to account for the spatial structure of the data when you're choosing how to iterate through it. So for example, a typical way to analyze larger than memory data sets is to break it into chunks, analyze one chunk at a time, and then combine them all together to get the results. That's going to be a challenge with spatial data because you have to make sure that all of the neighbors are present in each of the chunks if you actually want to do those computations correctly and at full resolution. Choosing the number of segments was still a challenge. So we have this sparsity to kind of help us guide us along, but it still doesn't actually give us a deterministic answer on what is the most appropriate number of segments. So these are some of the challenges that were still present. And so some of the optimizations we've introduced since then were to improve the scalability by separating out the spatial computations from the non-spatial computations. These are optimizations that I hope are relevant to anyone working with spatial data, especially from a machine learning side of things. So we pre-compute spatial information whenever we can. Any of the expensive computations should be independent and then later spatially refined. That simplifies the computation significantly. We want to minimize the total number of passes over the entire data set if that data set is too large to fit into memory, or if you have to move it to a GPU or something like that. And we also introduced warm starts. So if we have to fit multiple models, which is usually the case here because we want to try out different sparsity parameters, using warm starts will make those multiple models fit a lot more quickly. So one of the challenges, as I mentioned, is the spatial dependency. So here you can see the spatially dependent terms, but the actual distance term, this is an ordinary, just regular Euclidean distance inside of all of this. So we can start off by just calculating ordinary Euclidean distances that are not spatially aware at all. That simplifies the computation. And then we can reintroduce the spatial elements to the computation afterwards. So that makes it significantly easier to break up the data into chunks, significantly easier to parallelize those computations over many computers or many cores if you have them available. Originally, we parallelized over different models. So if you're trying a bunch of different parameter sets, each of the models would be fit in parallel. That was convenient because it's easy to implement. It's useful if you're fitting multiple models, but it does not allow us to communicate or share information between those models. And if you're only fitting a couple models, it doesn't help you that much. So our other optimization is this warm starts and parallelization within each model. So we fit the most dense model first, and then use the output of that as input to each of the more and more sparse models. So this allows faster convergence for the downstream, more sparse models. And it also gives us better statistical properties for comparing between models, because now we can kind of consider these models as nested models. So some of the statistical properties are more favorable when comparing between models. And so this is the result of the newer version of the algorithm. The actual algorithm is the same. The implementation has just changed a little bit. And so now we can actually use statistical criteria like AIC and BIC to kind of deterministically tell us which is the most appropriate number of segments here. So AIC and BIC, these are statistical criteria. The smaller they are, the better fit the model is to the data. These did not work very great before in the older versions of this algorithm, but now with the warm starts and nested models, they very nicely correspond with what segmentation appears to be the most appropriate one. So here's that mouse splatter as well. And I wanted to show a little bit of scalability. So I went on Pride and tried to find a data set with a large number of pixels. So this one had 845,000 pixels, which might not sound like a lot to many of you, but for mass spec imaging, imaging, that's a pretty decently sized image. If anyone knows any public data sets with more spectra, send them my way and I'll take a look at them. From a computational lab, we rely heavily on public data sets. So this is a segmentation of data set with 800,000 spectra, and you can see kind of how nicely that segments. So this was from proteomics of a human brain tumor. So this is a tumor. I'm not sure exactly what the details are beyond that, since I haven't taken much of a deeper look at the paper, but it's segmented pretty nicely and it was big enough to kind of make the size relevant here. So that's everything I wanted to talk to you about today. We have some future work going on in our lab. So one of our PhD students, Sai, is working on co-registration between mass spec imaging and other imaging modalities. We have another PhD student, Yunya, who is working on support for ion mobility in mass spec imaging. That's the end of it. So thank you, everyone. Thank you to the organizers for inviting me. It's great to be back here at Stanford. And I hope some aspects of this are going to be useful, even if you are not working on mass spec imaging specifically. Thank you.
Okay, we have time for a couple questions.
Thanks for the talk. I have a brief question about how do you get the ground truth of your segmentation?
Yeah, that's a good question. So the question is on the ground truth of the segmentation. And so I didn't really show much of the evaluation here. That's something that we do cover in the original paper back from 2016. And so certainly we can look at the morphology of histological imaging to kind of help us try to assess that. But what we used for evaluation in our original paper was actually some of these paintings. So here we have this image of a cardinal painting. There was another one in the paper of a farmhouse painting. And so the nice thing about these paintings, these were using DESI imaging, so it was easy to get the images of the paintings. And we could kind of neatly see whether our segmentation was able to pick up the different colors and reconstruct the original painting. Makes it a little bit easier for evaluation purposes than a biological sample.
Why did you get one from that
As for versus the histopathology images, so like I said, we could certainly do that to an extent. It becomes a little bit more challenging because we don't necessarily know if that is the ground truth when it comes to the biological distribution of different molecules. So if the brain and the liver share molecules, exactly how that should end up in the segmentation is a little bit less clear.
A great presentation. How do you address technical issues for batch effects and quantitation?
Good question on batch effects and quantitation. So this particular work does not really deal with that too much because this one is an unsupervised method. So the goal is looking at kind of a single image or a single sample. If we want to deal with multiple samples, we have other methods for things like statistical testing and classification. And there, of course, it is important to make sure that the data is properly normalized between different runs. And that is something that I think actually needs a lot more work in the mass spectrometry imaging community. So currently, normalization mostly relies on either TIC, total ion current, or root mean squared normalization, neither of which are supervised based on any kind of internal calibrants or references. And that is something that I think the MS imaging community needs to get a little bit more comfortable, including some internal calibrants so that there is a better normalization between different runs.
Great. I have a question sort of broadly related to this. I think one of the challenges for the non-computational labs is that the software stack is less developed for imaging in general and mass spec imaging in particular than sequencing or other types of bioinformatic approaches. What would be your general advice for the labs that maybe are not familiar coding some of this stuff themselves for how they can make use of the tools that are changing pretty rapidly?
Great question. So as far as taking advantage of the existing computational tools that are out there, so one of the goals with Cardinal is to make it easily usable by people who may not be initially familiar with coding. It does require programming to some extent, but we try to make that relatively easy. We have some courses. We have great documentation and tutorials, but we also have some short courses. So May Institute, we have a short course series in May at Northeastern. Cardinal session will be part of that, along with beginner and intermediate R. I think we're going to try to have some Python stuff as well there. And we are trying to get a short course approved for next year's ASMS as well.
Awesome. Well, join me in thanking Kylie for a really great talk. Okay, like I said, next up, we'll be hearing from Laura Sanchez from UC Santa Cruz, who's going to tell us about mass spectrometry applications for ovarian cancer.
Okay. Oh, should we? It's okay. I'll just do it. Okay, I'll go. Okay. So hi everyone, I'm Laura, not Angela. Thanks for going along with the impromptu order change. So I'm excited to tell you about something very different. I've seen a lot of talks today about tissues, and I'm not going to talk about tissues at all. So let's just dive in. In my lab, we like to study ovarian cancer. I think we all can appreciate that this is a problem. This is slightly out of date. It's now the 6th leading cause of cancer-related death among people with ovaries. And we know that it's typically caught late. And I think a contributing factor to this is that for the longest time, it kind of has it's not totally properly named. So in high-grade serous ovarian cancer, we know that the primary mutations actually occur in the fallopian tube. Those are the cells that move to the ovary and form tumors, hence ovarian cancer. And then in other types of cancers and this cancer, we know that this movement is really specific, right? So if we think about this is really an open channel. Why are these cells moving from the fallopian tube to the ovary? And then the most common secondary site is then the omentum, which is the layer of fat, everyone has over their intestines. And when you think about other cancers, we know that they have this really specific movement. And earlier, Benedict has already kind of primed us to the idea that in nature, we know that microbes move around by sensing things, and they're excellent producers of chemistry that they use to do this. And so when I started my lab, I was like, can we apply principles from chemical ecology to study human disease? So that's one of the things we want to try to ask and answer is, why do these cells have this specific tissue preference and movement? And is it dictated partially by chemical gradients? And so when we started out doing this, we basically had to come up with a different model system, because I want to highlight, we want to study things before you get a tumor. So I think once you have a tumor, right, there's all these like, treatment is terrible, things are nasty. We want to understand before you get a tumor. So how do we do that? How do we set this up and study it from a spatial context? So I had a very talented first graduate student, Catherine Zink, that actually took this challenge on. In case you're already being like, Laura, why don't you just get human ovaries? Those are really hard to come by. We only get 2 and they're not, if you lose them, that's not great. And then the other thing is we're working in a murine system. So murine fallopian tubes are called oviducts, and they're only called fallopian tubes in people. So anyway, we take 16 to 18-day-old mice. We're going to take their ovaries. So we're going to use healthy ovaries. And then we're using an immortalized murine oviductal epithelial cell line that has a P10 mutation. As a chemist, I was like, I don't know what any of this is. And I had a fantastic collaborator, Joanna Burdett, that's taught me a lot about cancer biology. And in her system, we know that P10 normally regulates AKT. So if you get rid of P10 by having these shRNA cells, you get runaway AKT, which leads to cell proliferation and tumorigenesis. So we know that if you put this cell line into mice, they will get ovarian cancer. So it's a tumor-prone cell line. So again, we want to try to study what happens before you get a tumor and the chemical communication there. And so to do this was actually a decent amount of work. And instead, I'll just show you a little video. So what you're going to look at here, if the video plays, this is always a risk, there it goes. We take these 8-well chambers that they normally use for microscopy and simply re-adhere them to the indium tin oxide slides that are normally used for MALDI imaging. And so what we found is if you use 2% agarose, you can put ovaries, so those are the little they look like little raspberry seeds. Those are ovaries that we are going to surround with a 3D co-culture of our tumorigenic fallopian tube cells in 2% agarose. So we're going to allow things to, communicate, gel together, and they're going to do this for 4 days. So we incubate them just like normal. We've already checked everything is alive and viable in this time frame. So again, that kind of showed you what the setup looks like. And then I think because I clicked in the slide. Okay, and so just to reiterate, what we're doing here is we're going to incubate things for 4 days. So we have our healthy murine ovaries surrounded by these tumorigenic fallopian tube cells, incubate, and then we simply just remove that little chamber that I showed you. We cut the sides away, the whole thing lifts off really nicely, and then we dry the whole sample down. You don't want to put wet things into a vacuum, so the agarose has to be dried flat. And once we do that, we then spray it with matrix. So other people have talked about the sprayer. We still use a mixture of CHCA and DHP to do this. So we're going to be doing mostly positive mode small molecule imaging. And then we introduce it into either a MALDI-TOF or now a MALDI-QTOF, and then raster over it. So I think, one of the cool things that we've seen a lot of today is what these imaging mass spectrometry experiments look like. So I hope everyone can appreciate that we gather a lot of data and a lot of spectra in an unlabeled target-free manner. And then we take an overall computational average, and this is where we, again, generate these hypotheses. And for this, we're really looking at what signals turn on in our co-cultures. And the nice thing about using these 8-well chambers is we can come up with rigorously controlled experiments. So we try to control for both gene specificity and cell specificity. So some of the controls that we like to add into these experiments are, again, using this murine oviductal epithelial P10 cell line. So we know that this causes tumors in mice. We have a scrambled version of this. So again, that's controlling for the gene specificity. And then for cell specificity, we use murine ovarian surface epithelial cells. So we just want to make sure that we're not just seeing things turn on because we've put things together. We want to make sure that it is related to the fact that these cells form tumors in mice, and we do these with and without ovaries. So this is the kind of experiments that we like to do. And if you're like, what does all this look like? That's what it looks like in the dried optical image. So these things dry nice and flat, and the whole thing can just go into the mass spectrometer. And so a summary of one of the early things that we found. So this is just, you're going to be looking now at all these different experiments, and you're going to be visualizing these ion images of the mass to charge ratios as a high to low. So high will be white and pink, low abundance will be dark or blue. And so again, we wanted to see things that turn on in the co-culture of the ovary with the tumorigenic cell line. And we actually found quite a few things turned on. And I'm going to freely admit that these don't look as beautiful as the tissue images. But again, I think we're asking and answering a very different question. So I'm okay with the fact that this isn't quite as pretty as some of the other stuff people have been showing. But you can see here, we can see signals like, let me see. I wonder if I can just use this instead. That way everyone can see it. Okay, yeah, right. So things that are ovary associated, things that are missing from the ovary, or things that increase in abundance in specific co-culture conditions. So we were very excited to see this. And, we used to say, a week in the library saved you a month in the lab. I would say a day in the internet saves you a month in the lab now. So Catherine went to the human metabolome database and dereplicated or wanted to identify some of these signals. And it looked like M/Z170 was norepinephrine. And we were like, does this make sense? The answer was yes. So we went into the literature and found that groups at MD Anderson have actually spent a ton of time trying to contextualize this signal when they were screening stress-related catecholamines, of which norepinephrine is, and found that in the literature, it's known to increase VEGF and angiogenesis, cell invasion, chemotherapy resistance. It's, peaks before ovulation in people. And I think most excitingly, in a retrospective study, they found that women taking beta blockers lived 60 months longer when they had ovarian cancer versus those not. So I think there was actually a direct translational value to this because norepinephrine should be working through the beta adrenergic receptors, which beta blockers would work on. So we thought this was really interesting, but I want to also say that in all this literature we found, no one ever knew what the source of this was. And so that's where I think these imaging experiments are quite powerful because we could try to address that. And so what Catherine did was slightly redesign the same setup we'd been using. So she took one of these sterile pipette media basins, then cut it so that we're now cutting these cells or these little chambers in half. And what she's going to be doing now is plating the cells on one half of the little divide, letting it dry, removing the divide, and then plating the other half of the co-culture. So we know that mammalian cells don't have the right machinery to actually move through the agarose, but chemistry can still freely diffuse through the agarose. So this time we're physically separating cells from the healthy explant tissue, allowing the co-culture to occur for 4 days, and then taking our measurements to try to address which of these two things was actually making that signal that we saw increased. And so you can see this is pretty easy. They come out, 150 microliters, dries really quickly. So this was a really nice protocol for us that we still use to this day for how we analyze when we want to know the cell or tissue of origin for these. Okay, and then yeah, it incubates again. That's nice. So just to remind you, this is what this setup is going to look like. And when we analyze this, to us, what we felt we saw was that this M/Z 170, if this is where the ovary was, looks like it's headed towards the cells. So we think that this was nice evidence that the ovary is in fact the source of norepinephrine. And then the cells would be using norepinephrine for all those other things that everyone has found at MD Anderson. And that again, there is directionality to this kind of chemistry. So then the question became, was this also relevant in our hands? So we did a quick little invasion assay, and you could see this is not statistically significant. However, when we couple it with a P53 mutation, which is the most common in ovarian cancer, and you treat this with 10 micromolar norepinephrine, we do see an increase in invasion. So this aligns really well with all the things that other people had found. And again, we use this as like an totally imaging mass spectrometry guided for ending up at these results. And so the next thing you might be asking yourself is, that's all fine and well, but why is the ovary releasing norepinephrine? Right, is it the chicken or the egg? So this is what we set out to find, because we think now, if we think about cancer as multi-directional, we have one half of the picture, but what's happening to cause that in the first place, because the other cell lines did not do this. And so for those that don't know, so this is what we next wanted to do. I'm actually a natural product chemist by training, so I love bioassay guided fractionation to find, new interesting small molecules. So I was like, can we do bioassay guided fractionation, but on this human system? So what we actually started, since again, we knew that this signal would arise without physical touch, we took cell-free extracts of our tumorigenic fallopian tube cells, and then made different kinds of fractionation extracts. So we have a spin column to trap proteins. Then we took the flow through and partitioned it into lipids and polar compounds, and then media. And this time, instead of doing bioassay, because the bioassay readout would be production of norepinephrine, which was actually really hard to test, we actually went back and did another imaging mass spectrometry experiment. So again, we're going to do the same thing with ovary, no ovary, but rather than using cells, we're actually just going to spot our extracts on top and see if something from the extracts will cause the ovary to release norepinephrine. So again, this is the optical image. And much to my dismay, because I'm a small molecule chemist, the protein fraction lit it up, and I was like, okay, now we're doing proteomics. But protein is too big of a word. So we ended up using these same spin columns and wanted to get at, which protein fraction is it? So we just did successive little spin columns to separate into like big, medium, small, very small proteins to see if we could narrow in what protein kind of class might we be dealing with. And again, we just use this simple readout of the mass spectrometry imaging to figure out which of these fractions lit it up. So you can see, just like real bioassay guided fractionation, we lose activity as we continue to fractionate things, which is not unsurprising. And it looks like we kind of lit up the 3 to 30 as well as the 30 to 50. So that kind of indicates that we're probably dealing with a protein right at the interface, because these spin filters aren't, super rigorous with what is and isn't at these cutoffs. It's not perfect. So we called our good friend Stephanie Colonna to do some bottom-up proteomics to see if we could figure this out. And again, since we're using conditioned media that have already gone through these spin filters, some of you might be saying, okay, 40 proteins is not much, but also this is not a crazy rich, it's not as rich as tissue itself. So we were really interested in, what proteins were specific to the P10. So we just did some quantitative comparisons. And again, the list is small. So we're looking at protein coverage, numbers of peptides, and we also took molecular weight into account. And so there were a couple of things that we thought were kind of interesting with like osteopontin and Spark, but we needed to kind of narrow this list down further. So while this was all going, we had a human cell line that we were also working with called SCOV3. We're not a super huge fan of SCOV3, but a lot of people use it. And it turns out it has the same effects with norepinephrine. And it actually lights these cells up in a totally different way when we screen it against a different panel of cell lines, human cell lines with ovaries. So SCOV3 is definitely very different. And we saw norepinephrine there. And when we fractionated it, we had the same thing. So what we wanted to use was the human cell line as a cross-validation for how to narrow our proteomics list, as like confirming which protein we should be working on. So when we composited all these results, it turned out Spark was still the winner. That was one of these three. So one that overlaps with the P10 and the SCOV3. A student in Joanna's lab, Tova Bergstein, who was very talented, checked the mRNA levels, and we found that indeed Spark was also increased. This protein, and I was like, okay, what is Spark, right? That's probably what you're all wondering. It's secreted protein, acidic, it's cysteine rich. It's right at that 35 kilodalton mark. And it's known to have interactions with extracellular matrix. When you think about the ovary, that's exactly what kind of surface you're dealing with. So we're pretty excited to see what did this do. So the first thing we tried was actually an overexpression of Spark. And it turns out Spark indeed does increase ionization of norepinephrine when we have this overexpression cell line. So that's great. So we think Spark is absolutely related to this increase of release of norepinephrine from the ovary. However, the knockdowns were not robust. So this is just showing what the knockdown looked like. We also tested this instead of the wild type, the P10 background, and didn't find a difference. So we actually think there's probably a functional limit of how much Spark we can actually get to be remodeling the surface of the ovary, because there's no change really between these two conditions. And when we tested recombinant Spark, we did not see this activity. So Spark alone is not good enough. So we think it's probably Spark working in combination with something else in the media. And then when we took the condition media and the recombinant Spark alone, that also did not do anything. So we're also not sure that the recombinant Spark, it may also require a PTM that we're not able to measure. But nevertheless, we're pretty excited to kind of fill this picture in that these tumorigenic fallopian tubes seem to increase their production of Spark, which is likely remodeling the surface of the ovary that results in the release of norepinephrine that then accounts for all those things that other people had seen in the literature. They just never knew where any of it was coming from. So I think this provided a nice opportunity to think about cancer as being a multipartite problem and not just trying to focus on fixing one side of it, but trying to think about how we can address the fact that multiple things are trying to communicate with one another in these systems. So again, this is where this is floating. And as I mentioned earlier, I also have a student that recently graduated that was working on what happens with the omentum. So the ovaries were all fun and great, but I think omentum represents something that's really common in other forms of cancer as well. So we wanted to know, could we adapt this system to study omental tissue? The answer was yes. I will say that each tissue does require some slight modification. So the omentum itself is quite fatty. So what we ended up doing is placing the omentum in the corner of these co-cultures and actually just cutting it off. So we're never measuring the omentum in these experiments because it basically does not dry in the same way that the ovaries did. But that's all fine and well. So this is giving us a good idea of how ionization changes in these different co-cultures. And again, we're using the same kinds of cell lines. So we're really interested in increases that occur when we have these tumorigenic cells next to healthy tissues. And so one that Hannah got really excited about was MRZ118. And she hypothesized that this was valine. So she did some experiments and we found that yes, it is indeed valine when we look at the MSMS data for these taken directly from this. And then the next thing she went on to do to quantify it was to do an ATRAC with SciEx, with SciEx's kit. And this is basically a heavy label for all the amino acids so that you can quantify it using these heavy labels. And oddly, she found that valine actually decreased in the co-culture condition when we were doing a quantitative experiment. And this, again, was totally an opposite to what we'd seen on this previous slide, where it looks like it's an increase. So we had to put our analytical chemistry thinking hats on and think about what was happening here. So when we went back and thought about this, the concentration of valine in the media is actually quite high. Instead of it, it looked like it had increased, but when we went through and just did a simple test of valine in the media, so we just did successive little dilutions here, we actually found that high concentrations of valine, you're getting ion suppression within the system. And once enough of it's gone, you actually see really nice ionization, and then it falls off again when you hit that threshold of not being concentrated enough. So I think this is a really interesting example, and one that we had never really thought of, that if you have metabolites in high abundance, we might be seeing them sometimes because they've been consumed. So if you have these high micromolar, low millimolar concentrations, especially of some of these endogenous metabolites, I think this is something that probably is in a lot of people's data. So that was interesting to us. So in the case of these tumorigenic cells with the omentum, they're actually consuming valine, and that's why we see it ionize. And so when we give the cells more valine, they actually proliferate a lot better. So I think this is really interesting to think about how we contextualize our results with what this might mean, again, in the context of cancer, because now we have an example where this is a media component that's impacting the phenotype that we can see and observe in the lab. And I want to just give another plug for things that are coming out the pipeline soon with the omentum data. We like omentum because we can better control this with spheroids, and it's much easier to get human omentum than human ovaries. So these are some of the things that we're continuing to work on, and we really like this system because it represents a plug and play. And again, we're trying to find chemistry before you get tumors to understand why specific cells move to specific places in the human body. And with that, this is a lab. We're just a little bit down south where there's redwoods. Catherine started the project. Hannah recently graduated, and Carly and Jason here are continuing. None of this would be possible without, the people that pay the bills. So thank you to the NIH, and I'd be happy to take any questions.
All right, just a reminder, if you're on Zoom, go ahead and type them in the chat and Candace can read them out. And for all of you here, please take advantage of the microphone.
Thanks, Laura. That was amazing. Also, I think you're the only person who really does these functional studies with the mass spec imaging, which is like kind of your field. Is amazing. So there's no need for tissue, I think. I was wondering, because I remember you have a timsTOF flex too. For the small molecules, do you use the ion mobility to improve?
Yeah, there are some projects. I think we found the ion mobility for some molecules that need it. It's great, but we don't. I think some of the things that Kylie brought up earlier is the data sets just become unwielding. So we turn it on when we need it, but otherwise we've just found that it makes the data way too big to handle. Mm-hmm. So yeah, we have some examples, like not in what I've shown you here, but there are some where it's critical. Yeah. And
maybe just one quick. You as a chemist using the mass spec imaging, it's known that for many of the lipids, you have sweet spots in terms of ionization and stuff that you really detect. But for the small molecule things, like the really small things that you look at, do you think, like, are there certain ostometabolite groups where you say, when you look at the data, they always come up? They're like low-hanging fruits for like small mass spatial metabolomics versus like some stuff you're just really hard to see?
Yeah, I think for us, it is still, so yeah, I think there aren't certain classes that we see all the time when we're in positive mode for the small molecules. But maybe I will say that I think once we know what an analyte is, like for the rest of the imaging, whenever we're dealing with norepinephrine now, we've switched to using the FMP10 matrix, which Per Andren at Uppsala has developed to try to enhance catecholamine detection. So kind of once we know exactly what we're dealing with, we do usually try to switch to something that gives a more robust readout. Cool. If that kind of answers the question.
Thank you. Yeah, amazing.
Yeah.
All right. Please join me in thanking Laura for a great talk.